So next we have up is Dr. Paul Huang. And just going to introduce him quickly. So Dr. Huang is the Deputy Director of the Royal Marsden Institute for Cancer Research Sarcoma Research Centre. It's one of the largest sarcoma research centres in Europe, and also he is the leader of molecular and systems oncology team in the Division of Molecular Pathology at the Institute of Cancer Research, London. So he uses systems biology and molecular pathology to study drug resistance in sarcoma and lung cancer. So he trained in Imperial College and MIT and was awarded the very prestigious Sir Henry Wellcome Fellowship in 2009 and the Cancer Research UK Career Establishment Award in 2015. Paul's published many papers, and most notably recent research efforts seen by Paul play an instrumental part in driving forward the world's largest sarcoma research database and helping to uncover what's considered as the Rosetta Stone of sarcoma protein alteration research to help understand and personalize treatment for people. So, having interacted with Paul previously, I know that he's a keen advocate for research, and he's always trying to bring together leading research to develop new ways to address challenges in cancer. So, without further ado, please could we welcome Paul.
Thanks, Rosie. So, if it's okay with you, I'm actually going to stand here because I think it's too close and I'm not as tall as Ed. So, first things first, so I'm the lab guy. And I'm the guy who's looking at all those complicated pictures that Ed was talking about, so don't hold it against me. So that's the first thing. Can you hear me? Yeah, perfect. Okay. So basically today I'm going to talk about research, and I'm going to talk about research in the lab, what we term as preclinical, so before the clinic, and translational research, which is research where we learn from, for example, patient samples and clinical trials. So I thought it's good to perhaps start with understanding what are the barriers to advancing research in leiomyosarcomas, right? So you need to understand the problem before you can tackle it. And as we already know, and Robin very eloquently said just now, our understanding of the biology of this disease lags behind many of the common cancers. So for example, breast cancer, lung cancer, prostate cancers. There are a few targeted smart drugs available, whether they be targeted therapies or immunotherapies, as M&A brilliantly talked about. And there's a general lack of awareness, right? Everybody knows what a prostate cancer is, a breast cancer, and you say sarcoma, and there's a big question mark. What is that even a cancer, right? So we in many cases, we are not really accessible in terms of the traditional funding routes. Oops, same problem. So what does this lead to? Well, one of the challenges, and if you see Robin at any of these big meetings like ASCO or CTOS, it's like a crazy man running around trying to get companies interested in running trials in sarcoma, because this is a big problem. It's not profitable for companies to develop new therapies. We have a big issue in terms of, you talked about just now getting tissue from patients to do research. There's a big issue of very limited experimental models, good experimental models for leiomyosarcoma to study in the lab. And that's how we actually test whether drugs are effective. There's huge diversity within the disease. Robin talked about anatomical side, but I'll talk a little bit about the molecular differences. And we've already had examples where trials don't recruit enough patients, right? So especially when there are multiple trials recruiting the same patient population, same leiomyosarcoma population, they're effectively competing trials and there's just not enough patients. And therefore trials take longer and you know, companies lose their interest and so on and so forth. So these are, I guess, practical challenges that we have to face driving research in this area. So I'm going to talk about three things. The first is biology and genetics. The second is touch a little bit on community-driven science, patient-led, for example, Count Me In. And then I'll tell you maybe my thoughts about where I think personalized treatment in leiomyosarcoma is going to be. So first of all, how do we go about understanding the biology of leiomyosarcoma and developing new therapies? To do this, we actually take a leaf from engineering, right? So we generate a parts list. So imagine if your car breaks down and you need to, you know, what's wrong with it? Is it the transmission? Is it the engine? Which part of the engine? So in order to understand this, what engineers have done is basically list every single part in a car, right? And then you will know when something breaks down, how do you fix it? It's the same thing with cancer cells. So here is one of those diagrams that Ed talked about. Each of these dots represents a part. And you have parts that, for example, regulate how well the cancer cell invades and migrates and metastasizes. We have parts that regulate uncontrolled cell growth. We have parts that, for example, control metabolism and differentiation. And so by understanding all these parts, you can start to get a feel for the biology of the disease. So how do we do that? We have a lot of different technologies to study different parts of biology. They can be at the DNA level for mutations. They can be at the RNA level, so DNA is transcribed into RNA, which is the messenger. And then they can be at the protein level, which is fundamentally what drives the biology within cells. Why do we bother looking at so many things? Well, the reality is that if you take a piecemeal approach, you're going to get this, right? So you're going to get your 6 blind scientists and an elephant. If you just take DNA or RNA or protein, you're going to think it's something else, right? So in order to get a holistic picture, we try to use a combination of different profiling approaches. So what I'm going to tell you today is a couple of different things. The first is different ways of looking at leiomyosarcoma, both from an academic point of view, so what we do in the lab, but also from a practical point of view, how is that going to impact patients? The first we'll talk about is DNA, genomics. What has genomics taught us about leiomyosarcoma? Well, we're very good at counting mutations. We count mutations and we are sequencing. We're really, really good at sequencing. The challenge is, what do we make out of the data, right? So this is a paper that came out from a good friend, Priya Chudasama, at the German Cancer Center. And she did a study where she looked at a large number of leiomyosarcoma cases and sequenced them like crazy. And she found basically the most commonly mutated genes in pretty much most cancer types, P53RB. So did we actually learn very much there? We learned that leiomyosarcomas have very, very complicated genomes. They have a lot of complexity and mutations, but there are also known frequencies in terms of high-frequency mutated genes. For example, 53, which is known as the guardian of the genome. It regulates many of the, controls many of the processes regulated with cancer progression, as well as RB, which is a known tumor suppressor that regulates cell cycle proliferation. So how is this information useful? This information is useful, for example, I'm giving you an example here in the context of therapy. So one of the very nice things is that sequencing can potentially be a biomarker for telling you drug effect or drug response. So actually a couple blocks down the road, where we do our research in the labs at the Institute of Cancer Research, many years ago, Alan Ashworth identified that cells which had mutations in this gene known as BRCA were sensitive to a class of drugs known as PARP inhibitors, and I'm sure Andrea will probably talk about that later. At the same time, Thomas Halliday at Sheffield showed basically the same result, that cells that had a mutation in the BRCA mutation, the BRCA gene, whether it's BRCA1 or BRCA2, were sensitive to these drugs. And actually, last year, the Science Museum had a mock-up to really demonstrate. This was actually not the actual, the replication of the actual experiment that led to the discovery. I'm not sure you can see all these little pink dots here. Those are cancer cells. And for the cells that were deficient in BRCA, you can see that the pink dots basically disappeared. So really the question is, BRCA mutations have been shown to be a good biomarker in breast, ovarian, now prostate. The question is, can we do the same for leiomyosarcomas? And the answer is yes, to a certain extent. So this is, again, another figure from Priya's paper. And what she showed, that if you look at genes that are in the BRCA and as well as related genes regulating DNA repair, DNA damage repair, you actually see some patients actually do have deep deletions and deep mutations in the BRCA gene. However, this is very histological subtype specific. So if you actually look at all sarcomas, so this is a study that sequence, again, we're very good at sequencing, sequence over 2,000 sarcomas, 45 different types. The frequency across all sarcomas is very small, only 1.4% of patients have a BRCA mutation. Now, the slightly good news is that if you look specifically at different histological subtypes, for example, LMS or soft tissue LMS, the numbers are actually much larger than, for example, some of these other subtypes here. So LMS is a good candidate for PARP inhibitors, and the good news is quite a number of different trials, either a single agent or combination, are evaluating the efficacy of this drug class in these patients. I should say that there's something to be said for none of these trials are using the known biomarkers for stratifying patients. So I think it's in part because, as Robin said, we don't know enough about the biology of the disease. So let's give the patients the drugs, and then we work backwards what the biomarker is later on. So the next thing is methylation. So what is methylation? These are little marks along your DNA that really form patterns. They regulate complex gene expression programs within the cell. And this methylation is particularly useful for a specific application in soft tissue sarcomas, and that's to address this problem here. So this is a plot really showing on the left-hand side initial diagnosis by a pathologist at the originating hospital, and then on the right, the final diagnosis by a specialist sarcoma pathologist at a expert sarcoma center. And you can see an LMS. 14 patients were actually misdiagnosed, right? And so instead of being LMS by this, diagnosed as LMS by the specialist sarcoma pathologist, it was actually something else, UPS, liposarcoma, or not a sarcoma. So you can imagine that given that treatment is dependent on what disease you have, if you have a misdiagnosis, this is a big problem. And of course, this really highlights that accurate diagnosis of leiomyosarcoma is still a challenge, and it's really the importance of being referred to a specialist sarcoma center, treatment center. Now, what the groups in Germany have done is shown that if you measure the methylation in cells, and this was particularly well in tumors of the brain, the central nervous system, you can potentially find patterns that separate out different sarcoma types, right? So what does this mean? So they decided to look at 65 different types of sarcomas, and they measured the methylation across their tumor cells. And what they found is that when they then plotted it on this map, you find that, for example, the LMS all the different LMS cases group very closely together, but they're very far apart from, let's say, synovial sarcoma or GIST. So in this way, potentially, you have a molecular way of classifying, accurately diagnosing what a LMS is. Now the question is, of course, this is theory. This is done in the lab. Can measuring methylation improve the speed and accuracy of diagnoses, potentially reducing patient anxiety and treatment delays? And of course, we all want to achieve this, but the reality is that when we start trying this, and the same group now tried it in 428 individual samples from a different center, only 61% of it was correct. 39 of the diagnoses were wrong. They were either wrong histological subtype, or they couldn't even classify it as sarcoma. So we still have a long way to go. We're still very far away from using methylation approaches in the diagnosis of leiomyosarcoma, but I think we're moving in the right direction, especially given that, perhaps there is I discuss with Robin all the time, the manpower challenges of having, enough sarcoma, especially sarcoma pathologists to look down the slides. Maybe this could be one way, particularly in, for example, developing countries. So the next thing I'll talk about is RNA. So RNA is the next step. So DNA becomes RNA. What has it taught us about leiomyosarcoma? And here's where I'll say that we stand on the shoulders of giants. One of the most early studies was really seminal studies published by Matt Van der Rijn in Stanford, where he showed really performing the first RNA analyses of leiomyosarcoma. And I'll talk about the data in a bit, but this was published in 2010. He followed up with another study in 2015 in a larger cohort. And I should say that now these samples comprise, I believe, the biobank supported by the LMSDR. Following on from that, a large US group funded by the NIH did a comprehensive analysis which included quite a number of leiomyosarcomas. There was the paper I talked about from the German Cancer Center, Priya Chudasama. Matt Hemming then published from Dana-Farber a study, and then most recently Rebecca Gladdy's group at Toronto published a study. And basically the nice thing, as I said to address the question just now, pretty much they all have the same result. So it was duplicated in different countries, different centers, different cohorts. So this gives you confidence that the result is real. So what is the result? most early studies showed that the tumors can be split into 3 groups: the red group, the blue group, and the green group. You can see that the details don't matter, but you can see that the patterns are basically different. And similarly, when he went to the next 2 studies, the larger cohort as well as the cohort by Dana-Farber, you can see quite nicely that there are 3 different groups. Here it's red, green, black. Here is blue, yellow, red. They call it slightly different things, and that might be personal preference. But fundamentally, all the studies have found 3 groups. So that's a great thing. We know there are 3 different types. So leiomyosarcoma, in addition to the complexities of where the disease has arisen, molecularly there's 3 different types. Unfortunately, it's really nice for biology, but we don't know what to do with it clinically. And this is one of the big problems. It's really not just doing everything in the lab, but doing the bridging from the lab into the clinic, right? And that's where we struggle a little bit in this space. Now, if I were to give you an example of where RNA profiling can be applied in the clinical setting, there is an example here. So what is a biomarker? What is a biomarker? So you go and see Robin. Robin will go and take your diagnostic specimen, and then we'll run it and measure the RNA, and then you put it through a computer, and it will shoot out 2 outputs, right? RNA profile A, it can be good outcome, for example, following surgery, lower risk of relapse, or good outcome following giving a particular drug, or poor outcome, in which case you may want to consider alternative therapy or more aggressive therapy. So what has this been used for? So I think the most advanced, still not clinically used yet, but the most advanced that's out there is this tool called SynSarc, which is basically a collection of genes, which I'll talk about in a bit. But what the Frederick Schibon, when he was in Toulouse, he did was to demonstrate that you can basically use this group of genes to split patients into 2 different groups. One in blue, which is C1, which is the low-risk group. So these are the patients following surgery have a lower risk of relapse. And this one in red here is the high-risk group, so the patients who are at higher risk of relapse. So what are these magical genes that can tell you, predict what these things are? A lot of them regulate the proliferation of tumor cells, so that's perhaps not surprising. If you have higher level of these genes, you are probably more aggressive disease and more likely to relapse. But some of these are actually biologically quite interesting. So these are genes that regulate chromosome instability. So whether your chromosomes and your DNA actually stay intact or they start falling apart as these cells duplicate very quickly. I would argue that biologically, we still don't fully understand how, for example, some of these genes regulate sarcoma biology. But as a tool, I think it's fairly credible. I also want to highlight the challenge that we face whether trying to move drugs or biomarkers from the lab into clinical implementation. And maybe a mark at the back from PTC can tell us the challenges as well. But the SynSarc algorithm was published in 2010. And the first trial that we're actually now just seeing conducted by the French sarcoma group is 10 years later. And this is not a drug, right? This is really just a gene expression profile. So the French sarcoma group is running a study where they're stratifying patients by their risks, by their SynSarc risk, where there's a high-risk patient, and they would give an experimental treatment which is more aggressive, and then the low-risk patients will get standard of care. So we'll see whether this pans out. But I think it's the one that's the most advanced so far in terms of moving a molecular biomarker into the sarcoma space. So that brings me to the project that LMSDR is funding in collaboration with our group as well, ICR and Royal Marsden, with McGill University and Stanford, to look at creating a parts list to tackle drug resistance in leiomyosarcoma. So I'm sure many of you guys are familiar with this. This is a bit outdated, it's from 2011, but to be fair, I think we've only got maybe 1 or 2 more drugs, so it's not that outdated. But in the context of the first line, we have doxorubicin or Gemtex, and then so on and so forth in the subsequent lines. So really the question is, the challenge that we face, at least what we study in lab, but I think clinically as well, is the majority of tumors ultimately develop resistance, so the drugs don't work anymore, and then the tumors relapse. So what does that actually mean? So what does it mean to become resistant, right? So to try and illustrate this, I'm using something that all Londoners are very familiar with. I'm sure you guys have seen this. This is our tube map. Sorry, it's a bit London-centric for those who are not from London. And of course, we are here in South Kensington. So imagine you want to go to King's Cross to get the train to Edinburgh. There are probably multiple ways to get there, but the fastest way is to take the Piccadilly line. Now, of course, this being London, every weekend we're going to have engineering works. So what happens? They decide to shut down the Piccadilly line. And this is exactly what drugs do. So if you now replace South Kensington with cancer genes, you replace King's Cross with uncontrolled cell growth. What happens when you add a drug is you're basically shutting down one pathway and you're unable to go from point A to point B. Now the problem is the cells are very clever and there are many other ways to get from point A to point D, and that's basically what drug resistance is. The cell and its microenvironment finds different ways to overcome this blockade here in order to achieve uncontrolled cell growth. So the big challenge that we have is understanding how a cancer cell reroutes to become drug resistant and therefore tackle this and find what's the next line of treatment or delay or achieve durable responses with current chemotherapies. So in the context of this particular project that we're doing with Joanna Principal in McGill, we've taken 54 cases from the LMSDR biobank. Half of them are good responders to GemTex, the other half are not. We're doing multiple profiling approaches and using this to build a parts list of GemTex responders and non-responders. We will then evaluate two different things, try using this parts list to identify drugs that can overcome resistance in cell lines as well as tumor explants in our microfluidics device, but also to identify biomarkers that can predict patients who are at higher risk of developing relapse following drug treatment. And I'm happy to say that Joanna will be presenting a very nice poster with some exciting early results from this study at CTOS next week. And finally, this is much longer term, of course. Any findings from here, we work very closely with our clinical colleagues, people like Robin, but also, I mean, this is really a partnership, right, with patients, patient advocates, and we can't do anything much as we would like to think we can. Patient industry involvement is very important as well. And if all things line up, we can then develop clinical trials, and hopefully some of these things will move into the clinic. So with that, I want to move to the second section, which is community-driven science. And I think this is very important because we touched upon, this sort of siloed science, right? I'm doing something in my lab, and then someone else in Australia is doing something else, and then someone in Boston is doing something else. So does it really make sense? Is there duplication of work? And then we're working with very rare diseases, so everybody is struggling to get sufficient samples, so how do we try and work together? So the good thing is, in the last 3 or 4 years, in particular the Leiomyosarcoma space, there's been a lot of effort in community-driven science, and I'll give you some examples of this. So, first of all, what's the underlying premise of community-driven and patient-partnered collaborative science? The underlying premise is, given the rarity of this disease, building a digital hub of clinical, molecular, and patient-reported outcome data freely available for use by the community will hopefully fuel the next generation of biological discoveries, novel therapies, and clinical trials. And this is quite lofty, but that's the goal that we're trying to get at, right? So where did this really start? So this really started in the Count Me In program at the Broad, at Corey Painter, and the first two diseases were metastatic breast cancer and angiosarcoma. And angiosarcoma, and because Corey is an angiosarcoma survivor as well, this was really the template, if you will, of how this could be applied in the rare disease setting. So what is it? This is the website. You basically go in, it's pretty straightforward. You go in, you click, you fill out a form. Once you fill out the form, they'll send you all the paperwork and the tubes to put your samples in, and then it sends it back to the lab to do the analysis. And what's remarkable about this is it was entirely patient-driven, right? It was through Facebook, it was through social media, and what was achieved. And this is, considering how big the US is, right? The number of sites and patients and loved ones who contributed samples, clinical data, was phenomenal for a rare disease. And this was, as I said, purely driven by patients. And if you look at really the numbers, to achieve at that time, 2020, to achieve about just over 300 patients registered and surveys collected for this disease is really remarkable. And then the numbers always go down when you start thinking about tissue, so saliva, tissue, It's just, some of it's paperwork, some of it's just finding the tissue. But it's still pretty good. I mean, we're getting about 20% of cases with tissue specimens. And hopefully now it's much better because we've refined their processes. So has this actually led to anything that's helpful for patients? The answer is yes. So one of the first things that Corey and her team did was to look at the genomic sequencing of these tumors, and she found that angiosarcomas on the head and neck, face, and scalp have a very high mutational frequency in their tumors. So they're very, very mutated tumors. And this was a potential biomarker for immunotherapy or checkpoint inhibitors. And true enough, when they look back at the data, there were a couple of cases. For example, this patient here, angiosarcoma patient, treated with pembrolizumab, and basically 219 days, and basically has an ongoing complete response. So, and this is currently being looked at in larger cohorts, but I believe in the US it's actually probably approved, is it? I think
we have a project for Leiomyosarcoma.
Yeah, I'm going to get to that.
Yeah.
But I think for this drug, I think the US, maybe the NCI have actually recently got the approval for this. Yeah. So anyway, so that's a good sign. So the great news, as Sharon was saying, is that there is one for leiomyosarcoma now, right? And this is the if you Google leiomyosarcoma project, How Me In, it's exactly the same process. They've done much better now in terms of they've got a huge chunk of funding from the NIH as well as philanthropy. We can discuss about how to get the UK into it. We already had discussions with Corey when she was still with the Broad, but there are, as Robin alluded to, data challenges to manage. In addition to the Leiomyosarcoma project, there are other projects. For example, the LMS spore. So this was another big chunk of cash that the NIH is giving. So we're moving away from the rare disease boring. You're not going to fund it. Now we're gaining a lot of momentum. And here the LMS spore is a multi-institutional study led from Michigan, and really is again looking at genetics and genomics of leiomyosarcoma. And finally, over on this side of the pond, as Robin mentioned, we've set up through generous funding from Cancer Research UK as well as the funders in Spain and Italy, the Sarcoma Accelerator Consortium, which is 11 centers over 3 countries shown here, where really we're trying to understand high-risk leiomyosarcoma patients and how to either delay the risk, delay relapse following surgery, or at the point of relapse, what do we do? This is quite a complicated program. There are multiple elements, so we try to collect clinical data, pathology, radiology, and all the profiling data we talked about. We try to develop some of the patient models from getting patient specimens to use in the lab. And there is also, as with everything nowadays, data science and AI, so there's also an AI component. For example, developing apps for risk stratification for physicians to use in the clinic to be able to guide clinical management. So I would say it's looking very bright. The future of leiomyosarcoma research is looking very bright. There's a lot of ambition in the program, and I think we can only go upwards, which was not the case 10 years ago.
Correct.
Yeah. Okay. So finally, my own personal perspectives. What should we do? How do we get better with leiomyosarcoma? Right? We talked a little bit about treatment, localized setting, advanced setting, chemotherapy, immunotherapy. What should we do? I think, as we all want to personalize therapy to improve outcomes. I think Ed's presentation is a perfect example of that, how you can tailor local treatment for specific and individual patients. But the reality is that if you look, and this group looked at 570 clinical trials over 30,000 patients, and if you had a personalized treatment, by personalized I mean, for example, the BRCA mutation, you find the BRCA mutation, you give PARP inhibitors, so that's a personalized treatment. Your outcomes are much better than if it's not personalized. So logically, we should always move towards and try to personalize our treatments to improve outcomes. And the same is for leiomyosarcomas. And we have a lot to learn from some of our other cancer types where this has been successful. On the left here is lung cancer. I mean, like I said, we're very good at sequencing our tumors, right? So in lung cancer, majority are KRAS mutations, EGFR, so these are different cancer genes. And there are good drugs for treating these indications. So if you have this mutation, you get that drug. Similarly for GIST, gastrointestinal stromal tumors is another type of sarcoma, you can see the majority of cases are KIT mutations, and then there are PDGFR alpha mutations. And again, for many of these, there are therapies available, and we are looking at third line, fourth line treatment now. So you're actually managing the disease like you would be managing diabetes, for example, right? Long-term, durable response. So can we do that for leiomyosarcoma is the question, right? And so I've titled this perhaps a little bit controversially: personalized leiomyosarcoma care, myth or reality? Could we start building a similar landscape for leiomyosarcoma based on stratification by, for example, biologically driven mechanisms or targets, or emerging biomarkers for existing approved therapies? So let's do a thought experiment. This circle represents the pie of all the leiomyosarcoma patients out there. So what do we do? So I've talked about the BRCA mutations. So perhaps the BRCA mutations could be a way of saying let's personalize treatment for leiomyosarcoma patients with BRCA mutations for PARP inhibitors. M&A talked very nicely about the various immune hot biomarkers for potential immune checkpoint inhibitors. That could potentially be another biomarker once we understand a little bit more about the biology of the disease. But there have been also various studies now showing that for some of the common chemotherapies that we use in the lab, in the clinic, for leiomyosarcoma, trabectedin, eribulin, pazopanib, there are a number of different either genetic biomarkers or RNA-based biomarkers that we can use. So I think as we build up the biology of the disease, we understand a little bit more, we can start converting leiomyosarcoma into what we're currently doing as standard of care for lung cancer or for GIST. And so I think that's, at least in my mind, where we should be moving forward to. Now, of course, there are challenges in clinical implementation. This is particularly true for European countries where there's a lot of centralized healthcare. So you start to think about, is it cost effectiveness and feasible, given the backlog, particularly with COVID, to do it for all sarcoma patients? Do you want to sequence all sarcoma patients? Is that feasible? Is that too costly for the system? And then I told you there's DNA testing, methylation testing, RNA, protein. So what type of testing do we do, right? And there's cost involved with that. And then very importantly, when? Do we give it before surgery, after surgery, at relapse, first line treatment, second line treatment? All these decisions cost and add additional workload into the health system. There's issues, we're already struggling with backlog, turnaround timeframes for testing and reporting. Ed talked about multidisciplinary tumor boards, although sometimes you wish you were flying in that room and seeing all these discussions. But anyways, we now have to add another layer, another meeting, which is the molecular tumor board, because you now have to say, okay, look, the genomics person have to sit in there and say, okay, what mutations does it have, and what drugs are available, blah, blah, blah. So that's another meeting to have and another workload. And then it's all great to do all the science, and the drugs are not available, right? So then that's one of the biggest challenges, is that we can sequence our tumors extremely well, but we can't get the drugs access to the patients. So for example, a PARP inhibitor is a classic example. You can see a BRCA mutation, doesn't mean that the patient can get access to the drug, which is a shame. Yeah. So, I don't want to sound completely pessimistic. We are moving towards that. So here are a couple of studies. This is the top left-hand card, is the DKTK program, master program in Germany, where they are doing a lot of sequencing in rare cancers, including sarcomas. This study here is across the US, where they use the Foundation Medicine platform to do sequencing. And this is the French sarcoma group, where they're doing a similar thing with multi-sarc. And really the idea here is these pilot studies are to generate evidence on feasibility. Which sarcomas do we want to do it in? And what's the cost? And this will aid clinicians, regulatory authorities, as well as patients in terms of moving towards next steps. So just to summarize, I do think there is a role, so not everything's pessimistic, there is a role for precision medicine in the management of patients with leiomyosarcomas. The early data from clinical profiling studies and trials are encouraging, but they're also sobering at the same time. I think this example of all the immunotherapies is a classic example. So you go in with all the best intentions, but the output is not what you hope you would get. And so, for me at least, being the lab guy, is that we need to constantly push our understanding of leiomyosarcoma biology to be able to bridge the gaps between biological mechanisms, biomarkers, as well as therapies. And so with that, I thank you for your attention, and always happy to hear you.
Thank you very much. Do we have any questions? Paul, you can keep hold of your microphone and I'll pass this one out. Any questions from anyone? No. So I had a question, which was if you were to prioritise what would be first to do next, what would be your priority?
I'm scrolling through the list in my mind. I mean, personally, Robin and I discussed this a lot. I mean, it's not feasible to sequence all patients. I mean, this is fundamentally impossible. I do think there is an element here of a lot of work trying to figure out. So going to the part of the negative trials. We've run a lot of trials, but I believe we've not learned enough from those trials. There are always a subset of patients who do very well on these drugs. We just don't know how to identify them. So Dr. Rubinson is a classic example. There are some patients who do extremely well, long-term responses. We can't tell them in advance who they are. We don't know why. Imagine if you could, going back to my pie chart again, you could then start to stratify which patients to give which drugs. And for me, that's like the biggest unknown. We're giving all these drugs, standard of care, failed trials. Perhaps we're not learning as much as we could from some of these studies.
Is there a role for AI in the...
Oh, yes. There was somewhere at the top of my list. Yeah.
Okay. Well, yeah, there will be somewhere, won't there?
Yeah. So if I go back, I didn't have, well, Ed stuff, there's a lot of AI stuff, but he didn't touch on that. So and a lot of the radiology is a classic imaging problems, it's a data problem. So how do you, by collecting a lot of these scans, can we interpret, for example, which is a high-grade tumor without taking a biopsy, or is there a Leiomyosarcoma versus a liposarcoma without doing a biopsy? So that is definitely, we're doing that. But there are other cool things as well. So for example, we're working with a guy in Spain who is doing all these nice computational models, so trying to say, can we put the cells, all these pathways that Ed talked about, into equations, and actually use mathematics to try and predict, okay, if you shut down, I don't know, gene X, cancer gene X, what is going to be the outcome? So we're doing that kind of stuff as well, and as I mentioned, both on the radiology side, but as well as the pathology side, because these are imaging problems, in some ways they're already been solved with, methods in other engineering fields already. We don't need to reinvent the wheel. We can just use the deep learning methodologies, the regression methodologies that are already developed in chemistry and engineering in this proposal. I think one of the big problems, and I think I can discuss this way better than me, is that the outputs from AI are a lot of them are black box, right? So we don't necessarily, it's called explainability. So we don't really necessarily understand. For example, if the black box says it's a leiomyosarcoma, how did it arrive to that decision? How does it relate to, let's say, when Ed looks at the scan, he's looking for calcification or whatever, it just says it's a black box. And if you don't understand, my view is if you don't understand the underpinnings of it, it can potentially be quite a dangerous tool. So I think that there are two sides there.
I think it just relates back to the question before this one, really, where you were answering what would you do with the money and so on. And you were saying about there not being enough feedback, really, I think, on your experiments or whatever you want to call them that you do. I know what I want to say, but I'm not sure I'm saying it properly. What is it that you need from when you do these trials?
Yeah.
What is it you're not getting? And do you know why you're not getting it? And do you know how? I'm sorry I'm asking lots of questions,
No,
but
it's fine, yeah.
yeah, why is it? What is it that's missing? Because there are frustrations from patients. I've just had a chat with people outside that we've got so much information that we want to give you, but we've got nowhere to go and no one to listen to us.
Yeah.
So what, that we feel we could help you by giving you feedback. Yeah, like today we've gone round the room and we've seen some people have stood up, years later. Why are they standing up and not, and why do there are other people who, like myself, probably never come to anything like this because they probably think, well, what benefit do I give? But they give a lot of benefit to sufferers,
So I think there's two parts to your question, two answers to your question. The first is, going back to Robin's point of, there's a lot of moving parts. So every trial, every study, there are always challenges. So some trials we just don't collect tissue because for pragmatic reasons like cost, ethics, some of these trials where you're running in multiple countries, it may not be feasible to deal with all the collection and shipment. So there are some practical issues, and those are on a very study-to-study specific. There's always resources. I'm never going to say resources, whether it's time, Robin's time, money, space. There's always resources, and rare diseases are right at the bottom of the list. Right, so I could write everything down, but it's going to be a very long list. But the second part to your question is, I agree with you. I think we need to do a better job in general of having patient feedback in informing the design of both trials and research studies. Right, so having a PPI forum or actively, we used to do this when the NCRI, which is the National Cancer Research Institute, which basically closed down now. They used to have a dragon's den for patients. So dragon's den, I don't know if they have it in the US, they call it Shark's Den.
Yes.
Yeah, so Shark's Den, yeah. So basically they have a patient panel, and you present your idea, and you just deal with the whatever that comes back, the feedback. And that's immensely helpful, because then you then feed that into the study, right? Are we is it too many times asking patients to come in to collect specimens? Is it feasible, to draw, I don't know, 500 mils of blood, right? Those kind of questions. So I think we have to do a better job at that. So I don't have one answer for you, one thing, but
not a lot has changed, because it's the same. There's a lot of change here. I don't mean that. I mean, in a blanket statement, the systems get under pressure and there aren't resources and all that sort of thing. But we need to, I think maybe out the box of how other we do it, because I'm going to go back to Sharon.
Yeah.
She's done a marvellous job on her own pushing this out. I've watched her over the years, and she's done a marvellous job with reaching people. She's a very good people person, and maybe you need something like that. Maybe it would be done through Sarcoma UK. I don't really know enough.
So I had an interesting meeting with, let's say, a wealthy individual from the US. And so I was telling him all the challenges that we face with resource, exactly what I just told you. And his first response, guess what was it? You need to find a good politician. Yeah? And you need to find
we won't bother.
Yeah. So I think there is a policy element here as well. I would say, we are just not, people are not shouting out enough. I mean, more recently, the Chancellor of the UK, his brother had sarcoma, so there was a little bit of news around that. But really, there is an awareness piece here that so I tell you pragmatically how we fund our research, right? The pragmatic way is it's all piecemeal. Between Robin and I, 60% of our time is writing for money, right? So how is that a good use of our time when we could be doing something else, right? And the reason is because there's just not enough funding there, so we have to basically use piecemeal. We have to ask multiple funders to fund a piece of work. Whereas, not to compare, it's not us versus them, but if you did it was in breast cancer or in prostate cancer, there are these big funders, £1 million. I mean, the accelerator is 2.5 million. So that was a big investment by CRUK. Going back to Ed's diagram, how much, now if you replace it to how much funding did CRUK give, sarcoma wasn't even there. Now with 2.5 million, at least we're a little bar at the bottom. Yeah. So we're doing okay for what we are, but we could do even better. And I think it's awareness, it's patients have a very powerful voice. I wouldn't underestimate that. You guys are pushing the agenda for us. You guys are way more credible than me and Robin, honestly. Because we are always seen as we have a conflict of interest. We just want to do it to advance our careers, whatever. But patients have a lot of.
But we're individual.
The
problem is, I come to my, I'm sitting there, great group of people here, and I come here and I don't know what's wrong with the next person next to me or where they are in their journey, and it's difficult to relate, but maybe they can think outside the box and, there be someone at those clinics who can bring those people together who are happy to, help.
I think that's very good.
Because I'd like to, but I just don't know where to start.
I don't want to put Sam and Helen on the spot, but I think...
We need to speak.
Do you want...
Well, I can shout.
Okay.
But I'm good at that. Sarcoma UK do contact us because we do have a really good patient involvement group now. Some of the people are here actually who actually sit on our lay review and get involved in research and support these guys and that. So we are trying to build that body. It's about 130.
I agree, but what I'm saying is the place where you go for your treatment could be the hub of bringing that. I know that we get our forms and we get our things, but it's sort of like I haven't had any interaction because I don't need it, yeah? So it's sort of waylays a bit, you know, unless I do something alone. But it's just, I could come to you, you're quite right, and I do know about you, so it's just, what do I say when I come to you?
That's it. That's what you have to say. Just come to us. Just come to us.
I will tell you, sorry,
Please go.
I will tell you, in Germany, last year we start a patient academy, who patient can learn how I read sorry, my word, the reports and so, and they will have later patient, some patient from bigger groups. They look with the doctors and with the other people that they make better research for what the patient needs. So, and it's new, it starts new, and I think the German sarcoma group works with a lot of European groups together, and some of the universities very special for sarcoma, and they started in a sarcoma group. So I hope it will be better and better. That is, that the patients have Ergebnisse. Results, yeah, sorry. He's German too, so that the patient get results like new medicine or so, because now the research is often, everybody made small research for himself and sometimes the same, but without any research at the end for patients. So we hope it will be better.
No, I completely agree. I think, there's definitely a piece of work to be done here. I would say, speaking on this side, where we, as I said, we write a lot of grants, the funders now, whether it's Sarcoma UK or some of the pediatric or bone cancer charities, there is a very big element of patient involvement in design of your studies, even laboratory studies. So I think we are fully aware of that. And actually, I think Sarcoma UK, for instance, has now a patient representatives in their grant review panel as well, which have exactly the equal scoring as the scientific review. So
when you go for your treatment, you get a piece of paper, especially when you first diagnosed, you just get these leaflets all into you. Well, we all know what we do with leaflets, don't we?
Yeah.
They tend to get lost or thrown in the bin. And you don't really, it's not personalised. I think that's what I'm saying.
So
yeah, and I think I would also,
personalised.
sorry, if I could just add one, I think if you also flip it around, I always open this to, sometimes it's good, so, I'm in a somewhat privileged position, I can meet you guys and learn from you, and it's always a two-way process. I'm also learning from you all the time. But the people in the lab are in the lab, they're just sitting down looking at stuff. And I think it's very important, if people want to come and talk to the people in the lab, or, I used to do this for a lab away days, we invite someone from a patient or patient advocate of sarcoma, you can come and speak to the lab, tell them about your experience, why research, why treatment is important, all those kind of things. So I think it's a two-way dialogue, definitely, for sure. And I just want to reiterate, it's not one way. We are constantly learning from you guys as well. And it's just finding what's that middle ground.
So I think that patients need to learn from each other as well, yeah? And that's the hard bit, because we've not got,
So
in the hospital.
I guess this is why we have these forums, is to try so that we can learn from each other. But what I'm hearing is that people are keen to donate their tumour, for example, for further for research. How do we go about doing that? And if we wanted to be part of the Count Me In project, how do we go about doing that?
Okay, I think the Count Me In is we can go into the challenges of Count Me In, but let's deal with the first question because that's a more practical question. So how would you contribute tissue? So we're increasing the number of hospitals that we're adding to this study. We've now got, of course, the Marsden, Birmingham, Glasgow, Edinburgh, Newcastle, hopefully Liverpool soon. So we're adding the number of sites, and it's a straightforward consent. So in the context of this is in the context of surgery to collect the specimens and deposit it, and it can go through this pipeline. I should say that in general, most hospitals do have an archive as well. There is perhaps a piece to be done of trying to put together. So the way I do research now, if I want to study a very rare sarcoma, I have to contact 10 of my good friends from other centers in UK and Europe to be able to pull together 100 samples to do the analyses. I think we can do a better job at that, maybe in some centralized form. I don't know what the solution is other than picking up the phone at the moment. The Count Me In is a slightly more challenging problem because Europe has this thing called GDPR, and it's the bane of my life and the bane of Robin's life. It's an information governance document that basically says that, rightly so, patient information is controlled, and you cannot do this, you can do this, this is what you can do. But it's gone to the point where the bureaucracy is insane. And the challenge with moving data from Europe to the US is that in the US, basically, my understanding is there's a rule that says that the FBI can ask for any data they want, regardless. And many European countries are very worried about that point, and so we can't get over that legislation. And there's a lot of groundwork to be done if we want to get that. So data is really the big issue because the big underlying thing of Count Me In is the patient involvement in the patient data. And we can't overcome that barrier. So one of the things we talk about with Corey is whether we do two parallel studies, so the data doesn't cross. So we do the UK study and we do the US study, and then we compare our results separately. So like a test discovery cohort and then a validation cohort. So yeah, there are creative ways around it, but it's not so straightforward. It's not to do with the lack of willingness of the people involved, whether it's the patients, the doctors, the scientists. It's all to do with legislation and bureaucracy.
So, sorry, we have to finish for lunch now, but there's just one final question, which was, as an individual, if I wanted to do my DNA sequencing, how do I go about doing that? Do I have to do it privately? How does it work?
So Robin can comment from the clinical setting. All I would say is that from a research point of view, we have to be blinded. So I cannot know the identity of the patients. All the studies I talked about, the researchers are blinded for the right reasons. So that what Robbie can touch on, whether it's private care or for example, in the UK we are in a wonderful position. In some cases, you get your tumor whole genome sequence paid for by the government. There are some strings attached. You have to get frozen tissue and all that kind of stuff. But the key thing is that researchers are blinded to it. So you can't say, okay, I'm going to give you my tissue, and then I sequence it and give you back the data. That's not allowed for ethical reasons. But I think Robin can maybe address the sequence. Yeah.
Yeah. So I think the starting point is to have a chat with your oncologist about trying to do this through the UK-wide genomes project.
Sorry,
Can you hear me now? Yeah. That's better. Yeah, gosh. So I think the starting point is to speak to your oncologist about this and to see whether it can be done through the Genomes project in the UK. There are, depending on which hospital, there are ways of getting it done on the NHS as well. But I'd have a conversation with your oncologist about how best to get that done. I think in terms of doing it within a project like the Accelerator project, yes, because you've got hundreds of samples and all the information from that, so you can do it within a project like that. I think for an individual person, it will just be the readout. I don't see how AI can help with that. But within a larger project, I think it's very, very useful. Very, very useful.
Thank you very much. So if there are any other questions, then can we take it outside at lunchtime? So we are going to now break for lunch. There will be lunch outside. Sharon, what time do you want us to meet back up? Probably, maybe at 25 to? So if we can meet back in here at 1:35, then that would be great. And just a round of applause once again, and many thanks to Dr. Johnston and Dr. Huang. And you have a little something to say thank you.